Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This episode is with Kate Adamala. She is an assistant professor at the University of Minnesota. Kate is a renowned synthetic biologist and biochemist who has made significant contributions to the field of artificial life. Her research focuses on building synthetic cells, designing artificial genetic circuits, and exploring the origins of life on Earth. In this discussion, we will cover topics related to the first artificial life created in the lab, the creation of synthetic cells, various techniques for constructing artificial life, the origins of life, and the significant applications of synthetic cells. Enjoy the conversation, share and subscribe to support the podcast. Thank you for listening. Hi, Kate. Welcome to the podcast.
Hi, thank you so much for having me.
So let's start with the announcement from the year 2009, which was made by Craig Venter about the synthetic life, that we have made this one synthetic life or cell. And you work a lot on synthetic life and synthetic systems, how to build a cell. So let's start with that. Like, what did they create, and where do we stand on understanding a synthetic life?
So what Craig Venter and his team created was a synthetic genome that was then planted into a living cell, and then they replaced the genome of that natural living cell with that new synthetic genome. So it wasn't specifically a synthetic cell created from non-living components, but it was a cell created with a synthetic genome. So it's basically a living cell that has a genome that was completely made in the lab. And that was revolutionary because that was the first time we were able to create a living cell with a completely known genome, with a genome where every single nucleotide is made in the lab, synthesized in the lab, and we know the sequence of that entire genome. That was obviously a huge progress, not just to the engineering synthetic cells, but also to a lot of other areas of biology. That was really significant because, for example, one of the things that came out of this project was a new cloning method that everyone uses now. Dan Gibson was a postdoc at JCVI when he was working on building synthetic cells, and he developed the so-called Gibson ligation, which now everyone uses in molecular biology labs. So that's just one example how this research benefits other areas of work too. And the significance of that synthetic cell is also that this is the first time we have a completely known sequence of a genome of a living cell. But the issue is that we don't exactly know what all the genes are doing. There are several dozen so-called essential genes of unknown function in that organism. So we know they're needed for life because if you remove even one of them, the bug gets dead. But we don't know what those proteins are doing, what those genes are translated into. And so that's the shortcoming of that work, is that yes, we built a synthetic cell, but we still don't know exactly how it works. We don't know every every molecule, every gene in that cell.
And what is a synthetic cell?
That is a very good question. I think everyone who you ask is going to have a different answer to that question. To me, a synthetic cell is something that, a true synthetic cell is something that has not been alive before the experiment. So basically something that is made from scratch in the lab, that's kind of a true synthetic cell. There are obviously stages and kind of ways in which you can make a synthetic cell that doesn't fit that definition. The most important thing is that synthetic cells have to be controllable. The kind of a rationale for building synthetic cells is that you want to be able to build something that we can completely control, that we're actually engineers for a change, not just kind of studying things we don't understand, but actually are able to control what we're studying. And that's the key of a synthetic cell to me, is something that looks like a cell, smells like a cell, but is not living, complex, natural cell. And I realize that's not a very satisfying definition because everyone has kind of their own different definition. To me, a synthetic cell is something that is fully controllable and fully understandable and also living. And I know you're going to ask me for definition of life in a second. And so I'm kind of putting some insurance in place here. I don't have a definition of life. But once we build a synthetic cell from non-living components, hopefully the definition of life will become easier to define too.
Well, just to differ from that point, I was not going to ask you for the definition of life because I know that I have never received one from my past conversations. And I think more and more I think about it, it doesn't make sense to like categorize it from certain perspectives or at certain point. For example, I mean, we can talk about, of course, protocells and the origin of life point of view. But the other point of view that I really like is the fact when we become life or living. At some point we are just chemistry, which turns into sperm, a specific sperm, and an egg, which possibly meet, and we get an embryo, and which transforms into a human baby, and that's what we are, right? So at which point we become living, it's again a question. And like, how do you categorize it? It's so difficult.
So the question you were asking, there are actually two different questions there. One is the very difficult one to which I have no answer for, which is when do I become an individual life? But in the process of creating a baby, you're never actually creating new life because there's always life there. The sperm is a living cell and the egg is a living cell. So when they combine, you combine two living things into a new living thing, but you're not creating life from scratch. You're not creating life from non-living components. And in that way, every time you make a baby, you're not creating life from scratch. You're just making a new individual from other living individuals, but you're not making life from scratch. So it's not the beginning of life, as in there was something dead and now it's living. And that's one of the fascinating things about biology is everything you see comes from an unbroken line of life since the origin of life on Earth. Because there was never a point after the origin of life when a dead thing became a living thing. And that's the fascinating thing about biology is that all life all the way down. And sperm cell is alive, egg is alive, new embryo just comes from more life. And that's really cool for me.
Yeah, but that's the thing. Like, till the time we are using the same metaphors, we I think don't evolve in a way, because probably we can think of it in a way like when chemistry transforms into biology, and that definitely happens. Certain chemistry transforms into sperms, and certain chemistry transforms into eggs. And this biology, the living systems that we are saying, they mate or they combine, and they form an embryo, right? So
and that's fascinating.
It is indeed, yeah. So yeah. If let's say we understand kind of a synthetic cell, as you said, that it's this minimal system which we can control and understand, does it also have to replicate?
Yes, to me, that's my private definition is that a cell has to replicate. It's obviously a very flawed definition, because if you say that life has to replicate, then you are not alive because you cannot self-replicate. You need your wife to replicate. So by definition, that requires replication from an organism, you're not alive. And that's why I don't like to say life has to replicate. But that's the reality of the Darwinian evolution. In order to evolve, a life has to replicate. So I'm willing to say, yes, a synthetic cell has to replicate. It's going to be really difficult to draw the line at which point the replication is sufficient to really maintain evolution. But I think, yes, synthetic cell has to replicate.
Yeah, and so there is one more term out there. It's called artificial cell, artificial life. So what is the difference between the synthetic life or artificial life?
There really isn't. It's two different words used to describe the same thing. Synthetic or artificial is pretty much the same.
Yeah. So when we say that we want to build life in the lab, so what are we trying to do? What sort of life or living system we are trying to create?
That depends on who you ask. I would like to start with building something that looks very similar to a natural living cell. So when I say I want to build life in the lab, I mean I want to build a cell that's built from the same building blocks as natural life right now. And I want it to perform the same functions as life performs right now. So I want it to grow, to metabolize, to replicate. The kind of end goal, the long-term end goal, is that I would like to build life forms that are different than what we know as life, modern life. Because right now there is only one life form on Earth. It's the part of biosphere that we're part of. It's the life that came from the last universal common ancestor. And that's boring. That's just one life form. Like all of biology has been studying this one form of life. I would like to make different kinds of life. I would like to make life that looks different, that behaves different. But we need to crawl before we walk. So in this case, it means we have to first build life from non-living components that looks like our own modern life. And then we can move on to building life as we don't know it, to building new kinds of life.
Yeah, it's boring chemistry, biochemistry in a way, but it's like the end result is so fascinating. That means there is so much diversity and stuff. It's amazing, right?
Life is amazing. There's so much diversity, but on a biochemical level, it's all the same. Everyone uses the same ribosomes. Everyone uses the same DNA, RNA, and the proteins with the same amino acids. It's like when you're a biochemist looking at the diversity of possibilities, how many different possible molecules are out there that can support life. And then life is just limited to that small set. That's what I mean as kind of a boring, because there are other options. You said you talked to Steve Benner. I'm sure he talked about alternative genetic systems and how life can move away from those four canonical nucleobases. There's so many possibilities out there. And the same with amino acids. There are hundreds of amino acids that could build proteins, and we're stuck to using 21. And that's what I call boring. It's basically life haven't really explored this incredible diversity of biochemistry that's possible. And that's up to us, the people that actually build synthetic cells, is to try to build cells that do explore that diversity. And that would be amazing.
Exactly. Yeah. So what I get is that there are two approaches in a way when we think of creating synthetic cells, this bottom up and top down. So what, for example, the people at Craig Venter Institute, they did, that's more like top down, where you try to minimize the system to quite an extent and see how far we can go. And the other one is probably that you would describe, which is this bottom up. So what are these approaches? And do you think that one is better than the other, or it's just a way of approaching the question?
They're complementary. One is not better than the other. They're both needed, because one day hopefully we'll meet in the middle, having an actual synthetic cell. So yes, the approach that I'm using is the bottom up, which means you're starting with something that's not living, that's very simple, and you put it together to the point when it starts being living. And the Craig Venter's approach, the top down, it's the other way around. We take something complex and make it simpler. And hopefully one day we'll meet in this middle, which is a fully understandable living synthetic cell. We're not there yet. Neither of those approaches really resulted in a fully understandable, engineerable synthetic cell. But both are needed because we're learning more and more about life as we're doing it. And so hopefully one day we can make it, we can build it.
But for both the approaches, we do need biochemistry. It's not like we are using something else. We need this carbon, nitrogen, oxygen, whatever, like the basic elements that we have, based biochemistry. This is what you guys are using, right?
Yes, the biochemistry is at the bottom of all of it. We're essentially chemists trying to bridge that boundary between living and non-living.
Okay, interesting. So, and what are the crucial components? What are the major components that we need to create a cell?
So right now we're sticking to the boring. We're sticking to the existing modern biochemistry. So our synthetic cells have DNA genome that gets transcribed into RNA, and then RNA gets translated into protein. And most of the time we're using the same building blocks as nature does. So we're using canonical DNA, RNA, and canonical amino acids. And we're using the ribosome, natural ribosome. So all the building blocks are the same as in a natural cell. And then we put it all inside a phospholipid membrane. So it's the same lipid membrane that encapsulates natural cells. And then we're hoping that it will start exhibiting as many kind of functions of a living cell as possible.
Yeah, so you said that it's DNA, RNA, and ribosome, and this you encapsulate in a membrane. But do we need those compartments? Do we need membranes, or we can also use this non-encapsulated system, means just the compartment-free systems?
You can use the compartment-free system for sure, but it won't really be living because one of the hallmarks of life is individuality. The ability to evolve, to undergo Darwinian evolution, relies on the fact that there is self and non-self. So to evolve, you need to change in relation to the rest of your population. And that's only possible if there's a compartment, because that's only possible if you can separate your own genes from your neighbor's genes. And if you don't have compartments, then you can use that system for biomanufacturing, for making compounds, but you cannot use it for actual kind of living processes. And that's why most people in the field seem to agree that compartments are necessary. They don't have to be lipid compartments. Some people use protein compartments or even microfluidic systems. But it has to be something that allows you to separate small reaction environments, so kind of a small drops of biochemistry separated from other drops of biochemistry.
Yeah, I mean, I was referring to the systems like liquid-liquid phase separation, where
yes, exactly.
Yeah, people. I mean, at least a lot of people now, they are thinking that maybe these are important in a way that because
yes.
you can be in the same environment but still isolated from like certain other machinery and still act as an individual system.
Yes, a lot of work is being done now, including in my lab, on those liquid-liquid phase separation compartments. And it's funny because it's almost like it went a full circle, because there was a book on coacervates as thing that gave rise to life almost 100 years ago now. And the operant theory of coacervates gave rise to life was like one of the earliest scientific origin of life theories. And then people moved away from it. People moved away from coacervates. They turned to lipids. But now we're kind of going back to coacervates. We're going back to, well, maybe there's something to it. Maybe coacervates are actually the way to go. And that's really fascinating.
Interesting. So when you talk about this system, so what does the order matter? For example, so if we take, I don't know, certain DNA, RNA, ribosome, and lipids, if we just put it in the in a test tube in some solvent, does it form a synthetic cell, or what do you need to do to form it? Like really or what are the conditions that you guys are using?
So it has to be at the right pH. It has to be at the right concentration of everything, really. Lipids, proteins, the DNA has to be at the right concentration. So there's a relatively narrow set of concentrations at which it works. And most of the time we make the guts of the cell, so the cell-free protein expression system first, and then we encapsulate that in a lipid compartment, or sometimes in this membraneless protein coacervate compartment. But first you make the central dogma. So you mix the DNA with all the enzymes and small molecules, and then you want to encapsulate that. So that's usually the order of business. And in order to get the lipids to form coacervates, they have to be at the right concentration and at the right pH. And there's again relatively narrow set of conditions under which that's possible.
Yeah. So just one disclaimer here that we can give is the fact that it's not like creating the life from scratch in a way, where,
yes,
for
we're
example,
not
Lee
making it
Cronin
from
and
nothing.
yeah, people like Lee Cronin and the chemists, that they are thinking that they can really start from, or for example, Nick Lane or Bill Martin, that they are thinking that they can really start from gases and go till I don't know, biochemistry, and then finally get the life. It's not the case. We are just using, or at least the people who are working on it, they are still using these systems which are present but isolated, and then trying to put it together.
Yeah, the rationale is that eventually we want to get where Lee wants to go too. We want to use other building blocks. We want to use molecules that are not natural, but we don't know how to do it. We don't know how to make molecules to create living cell. So that's why we're using just existing complex natural molecules, because first we want to learn how to do it. We want to figure out how does one go about putting together molecules that then eventually will start living. Once we know how to do it, then we can replace those molecules with unnatural molecules. And then that's going to make me happy.
Okay, great. So now we can go back to the process that you were describing. So DNA, as in like the code, if we just break down the whole thing into steps, so DNA as in the code, and then you are talking about ribosome, which is certain translation system. And of course, it needs some cofactors. The whole system, it needs some cofactors. That's what you are describing as small molecules, and also it's the metabolism that it requires, right? And then it's finally the compartmentalization, which will just encapsulate the whole thing, right?
Yep, correct.
So,
and all of those molecules and the small molecules are purified from natural sources.
Okay, interesting. So what about let's start with the code or information itself, DNA. What is there in DNA? Are there genes?
Yes, they're genes, and depending on what you want to do with your synthetic cell, they're genes for different things. So for example, in the most complex, larger genomes that we worked with and other people, it's about 80 kilobase pairs, and they're all the genes that are required for all the cofactors for translation system, and then some polymerases, so RNA polymerases, DNA polymerases, and also genes that are required for formation of ribosomes and tRNA. And that's the biggest, that's the absolute biggest. A lot of the time, we don't have a genome that's that big. We just have genomes that are smaller, for example, genes that encode for membrane proteins, for antibiotic resistance. So it all depends on what you want your synthetic cell to do.
So 80 kilobases means like how many genes in numbers?
About 35 to 40 genes.
Okay. I was just asking because before we mentioned the work which was done at Craig Venter's Institute. So this minimal cell has about 473 genes.
Yes. Yep.
And so just to get an idea that, okay, how far we are going and till when we can make the system work. And so it's 35 to 40 genes, and it's like, it's not like, so these cells, they can survive then, or how does it work?
No, they do not self-replicate. That's the key thing. They replicate, but don't self-replicate. So they grow, but we have to induce replication. While the Craig Venter cell grows and replicates spontaneously. And in my personal opinion, that's the difference between living and non-living. Ours is not yet living because it doesn't self-replicate. And a Venter cell does self-replicate. There's been work published from JCVI by John Glass and his colleagues that estimate that the truly minimal, absolute minimal possible genome would be, on the order of around 200 genes. And those couple hundred genes, that means it's a translation system, so all the ribosomal genes, and the most basic metabolism possible. So just a few energy regeneration genes and lipid synthesis genes. And we'll see where we get. until we build it, we can't really say that that's the minimal possible. But I agree with it, those estimates. I think we are about halfway there from both ends. From the bottom-up system, we're getting to the point when we have about half of the genes that we need. And from the top down, they have about twice as many genes as they need really to get a minimal cell. So we're approaching the same middle ground target from both directions.
Yeah, although I know that after removing these quite number of genes, their replication system, it slowed down. So
extremely, yes. I work with those simplest mycoplasma-based minimal cells. We have SYN 3.0 and 3.1 in our lab right now, and they grow so slow. It's ridiculous how slow those bugs grow. It's almost like they don't grow at all. You wait and wait and wait, and a lot of the time your cultures mold before your cells amplify because the growth rate is that slow.
So how slow is it? What is the timeline?
It takes 4 to 5 days to cover a plate, which, it's ridiculously slow by standards of bugs.
Yeah. So, I mean, E. coli, it doubles in 20 minutes, just for the reference.
Yes.
People, right?
Yeah.
Okay, that's interesting. So, but do you think that it may just have to do with because this is also interesting, like if it already slows down and even if, let's say, the system survives with about 200 genes, but if it is slow, like, if it is, I don't know, the doubling time is a year or something, how we will monitor or how we will even get to know whether it's like it can do it, or we'll have to run those long experiments to make sure that it does it at some point, right?
Yeah, that would make for a really long PhD. I don't think that anyone would want to work on it. No, I think we will have to be really helping that synthetic cell at first. So we will have to be either inducing the division manually, like we do right now, or we will have to speed it up somehow, grow it in an extremely rich media, add all the components that it needs, because it's just not possible to wait that long for a division event.
Yeah, sometimes when we are growing bacteria in the lab and if they grow slowly, we just make a joke that probably we need to sing a song or something.
Yes.
It may help it.
Yes, exactly. That's the problem of any biological research is you have to wait for your bugs to grow, but then you also want to graduate on a reasonable timeline, and that's difficult.
Yeah, but so what do you mean by inducing the replication? How do you induce it?
So right now we induce it by pushing the synthetic cells through little holes. So there's a little filter that we literally squeeze it through, and that's called extrusion. And that basically forces them to induce division. And that's a manual process. They don't do that spontaneously.
It's basically a physical process.
Yes. Yep.
Interesting. So that means the components that it requires, they are completely missing, right? I mean, what else?
Yes. There is no components that would induce spontaneous division. So other people are actually working on it. There are a few labs, mostly in the Netherlands and in Germany, where people are working on spontaneous division systems, and they're getting really close. If you look at their work, they're almost to the point when you have those contraction rings and it looks like budding yeast. It's not there yet in a sense that it doesn't spontaneously divide the whole compartment, but I would bet you money that within the next couple of years, we'll see an actual spontaneous division of that compartment.
Great. Yeah, that will be also, I think it'll be good for the community to know what are those factors which are required to for the. So it's only for division, right? But
yes.
Then let's also talk about the transcription translation kind of system. So how does that work? Like, is it going with the same way, or like as it happens in the living cells, or you had to modify that system as well? So can you just take ribosome and cofactors and just put it with the DNA and like with this synthetic genome that you're using, it works, or do you need something else?
It does. So that's one thing that we did get to work already. It's you can take ribosomes, RNA polymerase, and put it together, and it does translate the genome. So that does work. It doesn't work as efficiently as if it was in a living cell, but it does work.
So what is the shelf life of the life of these synthetic cells? How long do they survive? Or
if you feed them and keep them happy, they can survive several days. Eventually, you're just losing the integrity of mostly ribosomes. They don't renew themselves as fast as they should.
So it's the ribosomes problem in a way.
Yes,
so,
right now we need replication of ribosomes.
So basically you have, let's say, a synthetic genome, and you isolate, let's say, these components from bacteria, just an example. So you lyse the cells,
Yeah.
you keep the cytoplasm, which will include the ribosomes and all the other factors, and this you incubated with the DNA, and you start encapsulating in the compartments. Is it
Yes,
like this?
That's correct. Yes, that's how we do it. And all those components are right now purified from natural sources, most of the time from bacteria.
Okay. I'm just trying to understand, like, what kind of slows down in this process. So if it has almost everything, is it the metabolism, or
yes, it's
what?
The metabolism is extremely primitive, as you can imagine. We only have energy regeneration system. Otherwise, this organism is a perfect heterotroph, so it makes nothing. You have to feed it all the small molecules, all the lipids, all the amino acids, all the nucleotides. Everything has to come from outside, and that's the limiting factor because that makes it really slow.
Yeah, and how the compartments are maintained. So, because what about the link between the information and compartments? Is there any link?
There is none right now. There is no lipid synthesis. So the only way in which membranes can grow is during the feeding process. The synthetic cell feeds on little liposomes filled with enzymes. So basically little non-living cells without their own genome. And the only way in which compartment can grow is by uptaking those lipids. So it basically eats lipids. There is no link between the genetic information inside the cell and the synthesis of lipids. Although it has been done. There's been work in the synthetic cell field by people like Neil Devaraj, for example, where they've synthesized lipids in a synthetic cell system. So there was that link between the genetic information and making the compartment.
Interesting. So, and then what is the kind of size requirement in this thing? Like, can they grow forever, or at some point they just
At some point they seem to stop growing, and we don't really understand quite why right now. In my hands, they grow till they reach about 10 micron size, and then they just don't uptake any more lipids. And I think that's possible. One possible explanation is it's because of the membrane stability. Once you grow too big, the membrane just becomes unstable and they burst. Although we haven't seen the bursting, but it's possible it's happening. So that's one kind of a limit, is that they cannot grow forever. At some point, they just have to start dividing.
Yeah, it's an interesting question for me, but it is a kind of technical question, because the size of these cells, that's another important aspect, right? Because when you may see bacteria with a really good microscope, like under really a good microscope, but is it possible? I don't think so that we can really see this synthetic cells under a microscope.
Oh, we can. No problem. The synthetic cells that we're working with are anywhere between 1 and 5 microns in size.
Okay.
That's huge by
huge,
microscopy standards. So there's no problem seeing them.
But that's interesting, because bacteria is like 1 to 2 micron, like at least E. coli. So they are bigger than.
They start out smaller, but then they grow and they end up bigger. And it's with good microscopes, not even super resolution, just regular good optical microscope, you can see features that are on the order of a couple hundred nanometers, 500 nanometers or so. is the smallest we can reliably image. So yeah, there's no problem imaging those guys.
Okay, I was thinking that maybe they are so tiny just because of the size itself, but this is interesting if they can still assemble in a way that like that you can see it under microscope.
Yes,
and what is the shape?
we totally can see them. Yeah.
And what is the shape of the cells then? Like, is it?
There's nothing fancy about the shape because we don't have cytoskeleton. So normally in bacteria and in other organisms, the shape is dictated by cytoskeleton, and we don't have that. So they're just blobs. They're round liposomes.
Yeah, and
they're
how
circles
do you?
Under the microscope.
Okay, so it's just like kind of vehicles and this one.
Yes, exactly.
Yeah, but you do introduce some transporters in the compartments in the membrane, right?
Yes, we do. Membranes have transporters, and most of the time those transporters allow for exchange of the nutrients. So from the feeding broth outside, nutrients go in, and the waste products from the synthetic cell are removed outside. Other people have also done active transporters. For example, people have demonstrated ATP synthesis across a synthetic cell membrane.
And then can it use like that ATP synthesis? Can it catalyze ATP synthesis or?
Yes. Yep.
Interesting.
It's possible to have that ATP synthase, and the ATP that they made then feed into the translation system, the synthetic cell metabolism, basically.
So did like at JCVI, did they study the some sort of cellular communication between these synthetic cells?
They do. It's not part of the original minimal cell project, but it's possible to study that communication both in the living mycoplasma JCVI cells and in our dead small synthetic cells. The cell-to-cell communication is actually really cool because you can study it on a really molecular level. You can see how those molecules cross between different cells. And we've done that with small molecules and with proteins and with nucleic acids, and it works.
So basically you use some sort of fluorescently tagged molecule and see how it is moving between different cells.
Exactly. And it's really cool because you can watch those interactions between cells.
So it'll be like some sort of flashlight moving between, two different areas or something, right?
Yes, that's a very good analogy. Yes.
Yeah. So now let's talk a little bit about like how we can use these synthetic systems. So what are like the major applications that you think we can use it for?
There's a lot of practical applications and a lot of foundational research applications. The foundational research applications are basically we want to understand life. And in order to understand it, we need to build it. That's the famous Richard Feynman's quote. That's terribly overused in our field, but it's still relevant. He said, what I cannot build, I cannot understand. And that's the main application of the synthetic cell engineering is if we cannot really say we understand life unless we build it from scratch. So that's the foundational research. On the practical side, everything you can do with a natural cell, you should be able to do with a synthetic cell, but with greater control. And that's really appealing when you think about engineering synthetic cells for bioproduction, for biomanufacturing, and for medical applications. We're using synthetic cells as those basically programmable, slightly smart bioreactors that let us make compounds that are then useful for applications. And for one example is synthetic cells have been shown to shrink tumors in live mice already. That's work from Avi Schroeder's lab. And he demonstrated that if you have a mouse with a breast cancer tumor model, injecting synthetic cells into that tumor shrinks the tumor. So but they can basically be used as artificial immune system, if you wish. Then there is a lot of applications in medicine. For example, work from Jim Collins' lab and Keith Pardee's lab, where they demonstrated that synthetic cells can be used to make vaccines, on-demand biomanufacturing of small molecules, including vaccines. That makes it really easy to imagine bioproduction on demand on small scale in places that don't have a cold chain. So in underdeveloped countries where you cannot set up a biochemistry lab in the field to make whatever you want to make, you can do it with synthetic cells because they can be lyophilized and stored over long periods of time. That also brings me to a really cool application that I'm personally excited about, which is space exploration. It's possible to use synthetic cells for building those kind of manufacturing systems in space. And that's another thing that we're doing in obviously collaboration with a lot of people, is trying to work on those systems so they can be used to make molecules under spaceflight conditions. Then another area of application is bioeconomy, biomanufacturing. Right now, a lot of the stuff that we make, a lot of the stuff that we manufacture is based on oil, based on petrochemicals. And we need to find a way to replace petrochemicals. And that can be done if you build a cell that can make those toxic chemicals for you. And it has to be done because we're running out of oil. So that's another kind of a very hot or relevant application field right now. And then there's diagnostics. A lot of, obviously now in the era of COVID, we're looking for cheap, fast diagnostics tools. And using cell-free systems allows us to build very rugged, robust diagnostics platforms. And that's another thing that a lot of people are doing. So there's a lot of applications. I'm obviously biased because I think they're all cool, but there's a lot of very practical applications.
Yeah, interesting. And the list goes on, right?
Yes, there's a lot of other stuff that's a little far out there, but still cool. For example, biocomputing applications, building those little biological computers, building kind of a systems that let us study populations. You're in a place where the Mendel did his first original work. We can do that work right now on a completely synthetic chemical system. That's what synthetic cells are. And we can do basically those hereditary studies. And that lets us study exactly where every molecule goes. There's a lot of kind of a cool applications like that.
Yeah, interesting. So let's start with the origin of life, or the fundamental research that you mentioned. So how we can use synthetic cells to understand origin of life direction.
The synthetic cells are essentially the proto-life. They're the cells, how cells could look like before the life started on Earth. And that lets us study those processes in great detail. That lets us study how life could have started, how life could have jump-started from molecules that we know life is using. That's the really interesting part, is that synthetic cells are made from the same molecules as natural life on Earth. And so studying synthetic cells lets us study those earliest molecules, those earliest life forms on Earth. And that also helps us imagine how life could evolve under different conditions, because you can recreate that prebiotic evolution using synthetic cells. And that helps us basically imagine how life could work if life started on Mars or on Europa or on Enceladus or anywhere else in a solar system or on an exoplanet. And that's a really powerful tool because it lets us basically recreate that prebiotic evolution.
Yeah, so once prebiotic chemistry gives us at least the biochemistry, modern biochemistry, from there we can kind of explore what goes on.
Yes.
Yeah. Meaning that how the smaller cells, some sort of protocells would evolve, and how those protocells can transform into something like bacteria and archaea, etc. Right?
Yes, there's a long way from a protocell to a bacteria, but every step of that had to happen gradually, and we can study those steps. That's why making synthetic cells is essentially recreating that origins process that happened on Earth 4 billion years ago.
Yeah, so here is another practical question, because at least when I think of origin of life research, I mean, one thing is it is an historical question, right? Like it's related to and there are so many factors. We need geochemistry. We need what sort of conditions were there and all that to know what or how the life will arise. But at the end, since like by training I am an engineer, and I think of it as an engineering problem more and more now,
Yes,
than thinking of it as like just the historical perspective, because we can have explanations, but those explanations we can only put to use once we can put the information together and kind of build a cell,
absolutely.
right?
Absolutely. It's not theoretical field of studies. We have to actually do it. We have to make it.
And this only this, until unless we kind of we will end up understanding that there is something major missing in our understanding of how biochemistry works,
I'm
or
hoping for that.
yeah.
I'm really hoping that once we make life, we'll realize that there was something big we were missing, that it won't be just like, oh, that's kind of neat but boring. I really hope it's going to be like, wow, I never thought about that.
It's like this revelation, like once once again people at JCVI, they created a minimal cell. They realized that there are 149 genes for which we don't know like what do they do in the cell. So if there is something like that we like a black box in the cell, that's a different thing, right? But at least we'll somehow figure it out then.
I'm really hoping that. I'm really hoping that once we build it, we'll be able to say, these are all the parameters and this is what was missing.
But on the other hand, with all the present technologies, I mean, we do metabolomics, we do, we can study lipids, we can do mass spectrometry in a way, like at all different levels. We can do sequencing. So we have I think all the components we have covered in a way, right? Like we have that kind of high resolution information at present times. So it's also difficult to kind of think, at least at this moment, what is missing in the system which won't do it. So
it's really difficult to think what is that we're missing because of all the knowledge we already have. It seems like we shouldn't be missing anything, and yet we do. There is something, because when you put those molecules together, they don't magically start life. If you ground up all the bacteria guts and put it back together, it doesn't recreate a bacteria. So there's definitely some kind of a biochemistry. I don't want to say it's any mystical life force or anything. It's just some biochemical process that we're missing at this point. And that's what's so fascinating about this work, because we don't know what we're going to find once we build it.
Yeah. So the other thing that you mentioned, that we can use synthetic cells to study life on other planets, for example. So if, let's say, I mean, what do you expect there? So of course, first of all, the conditions have to be the same. Biochemistry kind of have to be the same, what we get here. And this is where, again, prebiotic chemistry work, like the people that they are working on those topics, it's important for us to tell that, okay, like at this planet, uh like it'll be the same biochemistry which will evolve, right? And then we can say that, okay, it can be some sort of this system which will evolve. But how do you draw parallels? Like, do you have an example, like how really synthetic cell can help in astrobiology?
We draw those parallels by trying to recreate that evolution. So we don't have an example right now because we haven't found extraterrestrial life. But the problem about looking for extraterrestrial life is we don't really know what to look for, because we know that we can't look for just DNA, RNA, and proteins, because if we find that just means we contaminated whatever world we're looking at. We are looking for remnants of alternative evolution, for separate origins. And that's something that has to be completely biochemically different than what we know here on Earth. And that's really difficult. That's really difficult to imagine what can we expect to find. So building synthetic cells kind of helps us explore the possibilities. What happens if we combine particular molecules under those conditions? Like, for example, recreating how life could look like on early Mars, back when Mars was wet and had atmosphere. We can create those conditions in the lab, and then we can make those lifelike forms and then see, is it going to work or not? And if yes, then that's one of the possible scenarios. And okay, that's how life could have started on Mars. So that means we have to look for those particular molecules when we go and get samples from Mars. And that's just one example how that one day will be used once we get those samples.
Yeah, the next application would be if we continue in the fundamental research itself, for example, understanding multicellularity. That's another important aspect, right? So how we can understand that transition?
That's really difficult. Synthetic cells have never been made to demonstrate that transition between unicellular and multicellular. And I think we're still ways from being able to do that. People do those studies using natural cells, for example, the work of Mike Trevisano's lab, where they made unicellular yeast to become multicellular, the so-called snowflake yeast. They were created from unicellular organisms, and that's a way to study those transitions. We don't have a way to do that with synthetic cells yet, but we do work on things like ability of synthetic cells to communicate, as you mentioned before, exchange information between different cells. And that's the first step for multicellularity. You need to start having cells work together, work next to each other. And that's something we are working on, but we're still not there yet. It's still, quite a ways until we can say this actually was done and works.
So I did talk to William Radcliffe about snowflake yeasts, and this was fascinating that he could just apply, or they could just apply this one selection pressure, and from this they can get something like a multicellular organism, or it appears like a multicellular organism. Of course, there is another discussion, what is an organism and all that thing.
That work is absolutely fascinating because it shows you how biology really is biochemistry. Because you take cells that are not meant to do something, and then you evolve them to the point when some molecules are exchanged and it becomes multicellular. It's just fascinating.
Yeah. If you can show it in synthetic cells, that will be much more fascinating, because in bacteria so far, we know about biofilms and all that, but if it is working as an organism, this kind of example, I think it's not at least well understood or well explained in the literature.
Yes, absolutely. This is one of my dream goals, is to be able to show that the synthetic cells can evolve to the point when you can kind of give rise to multicellularity.
And can we also study eukaryogenesis then?
One day we will, but not right now. Those systems are too simple to imagine studying organelles. But there are people that are trying to do that, and they're having great success. People that work on claservates, on membranous organelles, have demonstrated that it's possible to have membranous organelles in cell-free system. So that's, half a step towards trying to get an organelle. So one day, definitely, we will have eukaryotic-like synthetic cells.
And how we can study evolution using synthetic cells?
We can either do artificial evolution experiments. So we can design synthetic cells that will have certain mutations, and then we select for those mutations. Or we can look at spontaneously risen mutations, but that's much more difficult because you need a lot of generations, and we don't have a lot of generations in synthetic cells right now. So right now, most of the evolution work is done using those so-called artificial evolution techniques, where you build a library with the mutations, and then you do selection experiments where you want to see which of those cells perform the best under certain conditions. So that's why it's called artificial evolution, because it's not like natural evolution works, but it's the same kind of a biochemical principle, is that you select for the genes that perform the best.
So, it'll happen only after you will have replication in the system, right?
Yes, we absolutely need replication in order to be able to say we can study evolution in any meaningful way.
Yeah. So the other application that you mentioned is or for example, in my previous conversation with Bill Martin, I mentioned that I see origin of life problem as an engineering problem, and he was like, "Okay, what will you do with that with that kind of system?" And the first thing that I had at the top of my mind was the environment issue. For example, that means we can create some sort of system which can do the job. It can be also like fixing gases, but also producing compounds or fuels that we need, for example, right?
Yes. That's the
So,
dream. That we have to do that in order to survive as a society. So that's a really big, crucial application of this.
but his comment was that why not to just modify the present organisms? Like
Yeah.
that won't be easy.
It won't be easy because present organisms are incredibly complex. We see that even in the JCVI work where they make this mycoplasma with a synthetic genome, and there's still over 100 genes that we don't know what they do. It's really difficult to engineer living organisms because they evolve with all this endogenous luggage. The remnants of billions of years of evolution, or those genes are there. And it's really difficult to introduce new functionalities, especially ones that are toxic. Like you want to make fuels, you want to make petrochemicals. Those compounds are really toxic, and no self-respecting bacteria will make that compound for you because they want to live. The bacteria are not stupid. They want to first and foremost live. So if you ask them to make something toxic for you, they won't just replicate and happily make it for you. They'll curl up and die. And that's the problem because the biochemistry is limiting them. The bacteria cannot change their own biochemistry. So that's why making cells from scratch and building new metabolism that's specifically designed to be able to withstand those harsh conditions, to make those compounds, that's probably a better way to go because we'll be able to design that metabolism specifically with the function in mind to do what we want to do.
But what if biochemistry has its own like chemical restraints? That means we can't go that far in a way. So how to deal with that?
And that's something we need to learn. We need to build those cells in order to learn that. And once we find those limits, I'm sure there are limits like that, like you mentioned, but once we find those limits, then that's the engineering part that kicks in. We have to design around those limits. We see that beautifully in other areas of engineering, for example, in aviation. Once we hit a limit, a technical limit, we design around it. We find new compounds, we find new systems, we find new ways to build planes that overcome physical limits that the old plane ran into. And the same with biology. There's no reason we can't do that for biology. Once we run into a particular limit, then we design around it. But it's only possible if we actually have a system that we can design. And that's the difference between natural cells and synthetic cells. You cannot freely design a natural cell because it's so complex. With a synthetic cell, you understand what you're working with, so you can design it the way you want it.
Yeah, when you listen to these kind of examples, again, you think of this engineering perspective. That it seems like that humans have really nice capacity in a way to think of innovation and engineering and like building technologies and stuff.
Yes, and we have not applied that fully to biology yet. That's the promise of the field. That's why I think people are right when they say this century will be the age of biology, because we will finally learn how to engineer biology.
Yeah, but also it was difficult if you think of, for example, biomedical applications, people have done a lot of engineering work there to quite an extent that if someone has a missing limb, we can have some prosthetic parts and stuff, right? But of
not
course,
made of biology though. That's the
exactly.
difference.
Exactly.
I would love to see those prosthetics made of actual wool, of biology, of living cells.
Yeah. So regeneration, or you think just to have like another part and kind of attach it?
I think of growing things. I don't think we'll ever be able to do regeneration in humans. We're just not designed for that. But it's possible to imagine that once we get better at tissue engineering, we'll be able to really engineer what's missing. So engineer things like limbs. I know we're far from there, but that's not crazy to think about it. bet you by the time the century is over, we'll be able to do things like completely regenerate an organ.
Yeah. So Nick Lane mentioned that he doesn't expect us to have a fully functioning cell in the lab, like sometime soon, according to him. I'm paraphrasing him, but I think it also depends like what kind of system he is thinking and stuff. But
absolutely, it's every time you tell scientists that something is impossible, that just motivates us to go and do it. And a lot of people do think it's crazy to imagine being able to make a cell, but you'd think other things that people called crazy would never happen, and they happened. Science really moves in leaps and bounds. Science is not linear. There are those progresses, those breakthroughs that happen. And then once that breakthrough happens, oh, it's obvious. We should have done it ages ago. It's once you do something, once you achieve it, it becomes really obvious that this is doable, and it always was doable. We just didn't know how to do it.
Exactly. And but it's also like how far we allow that craziness in the system.
That's another thing, is we don't really know how. We don't know how far, and we also don't know what is needed really right now, because we haven't done it yet. Once we do it, we'll be able to say, okay, this is this is where I draw the line. This is where the craziness happens.
Yeah, the other thing that we didn't talk about is is about viruses, for example. So viruses is a kind of system which probably you have. It's like some sort of synthetic cell without replication. So how do you look at this kind of like? I mean, viruses are so interesting in a way that. Where do you place them? They, of course, they create a list of issues when it comes to understanding life, living systems, etc. Or why not to, for example, just use a kind of the viral material that we have and try to just evolve replication there?
So viruses are not meant to self-replicate. They only can replicate with another living cell. And that's something that, in a way, you're right, that synthetic cells are like viruses right now, because they can only replicate if another living cell, in this case, me or my grad student, helps them replicate. And that's a very good analogy. Viruses are part of a biosphere. They need a biosphere to replicate, but they don't do it themselves. And I think it would be really difficult to build all of the machinery into viruses to let them replicate, because then you just have a cell. I mean, then you're not different than a cell. You're not a virus anymore. You're just a cell.
Yeah, your biochemistry transformed into biology, right? Like
exactly.
But the question is like, don't you think it'll be easy to just take, I don't know, the machinery of a viral particle and I don't know some parts of the machinery of host and just try to put it together and see if you can get a replicating system out of it?
My guess is that would be really difficult, because to have a whole system that replicates, you need to replicate the host as well. And at that point, it's just a cell replication problem. People have done viruses in synthetic cells. For example, Vincent Neuraud and Fritz Simmel's work, they both demonstrated that it's possible to make replication competent viruses and bacteriophages, for example, in cell-free systems and in synthetic cells. So we can use the synthetic cells to replicate viruses. We haven't actually built a synthetic virus like that, but maybe we don't want to, because we have enough gain of function research as is.
Yeah, interesting. And what about, like, since at the beginning we talk about this diversity of life forms. So again, when we use, of course, the same biochemistry, but there are at least few species where the biochemistry is in a way a little bit different, or at least the way it is used. For example, we can talk about the other unicellular organisms like methanogens and acetogens, and where the metabolism is completely different, right? Different in the sense that they fix carbon dioxide, etc., the other gases. So do you, or are you guys also thinking in a way to use a system like where we can fix carbon dioxide and like a reverse Krebs cycle and this kind of setup, rather than thinking of the canonical system that we have in most of the species which are present at the time?
Yes, definitely. There's a lot of work being done in that area, mostly with global warming in mind. And there's a lot of people that work on trying to engineer Krebs cycle and Krebs cycle derivatives, and as you mentioned, reverse Krebs cycle. One of the biggest kind of labs in this field is Toby Erb, and his work on engineering those carbon fixation pathways is really revolutionary because it helps us understand how that works on a molecular level and helps us build it from scratch. And I think that's going to be the way to do that. We're not doing it right now in synthetic cells yet, just because we don't know how to do it. But that's the dream. That's a dream to be able to do that one day.
But also in a sense that what if like because of course, if we think of from the historical perspective, for about 2 billion years, there were life forms that they relied on this system, at least because the oxygen, I think, came on the planet around 2 billion years ago.
Yep.
So, and if we just think of the time, they were quite successful if they were around for about 2 billion years. That's a huge time, right? So why not to kind of also think in this way, to have this kind of biochemical system and try to encapsulate that? Would it be too difficult, or what if like there is like it's probably much easier than having oxygen-based system, for example?
Definitely, and people are working on it. It is very difficult because we don't know the details of that biochemistry, but people are definitely working on it. And especially in the context of the origin of life, there is a lot of work being done in anaerobic environments on trying to do this primitive biochemistry before oxygen. And one of the biggest kind of areas of work is people are using iron 2 instead of magnesium as a cation in a lot of biochemical reactions, including ribosomes, because that's what was present on the prebiotic earth before oxygen. It was possible to have free iron 2 because there was no oxidation, and people speculate that that's probably what was the cation that metabolism was using. So there's definitely a lot of work on trying to reconstitute that.
Yeah, and so since you already mentioned about the iron +2, so because in the industry people use a lot of these kind of metal surfaces or to do the similar kind of biochemical reactions for many applications, to even like they're trying to produce biofuels, etc. So is there any evidence that kind of synthetic cells will have better yield in a way, or it'll be much better than like compared to those chemical processes?
We're hoping that's the case. We're really hoping that that will be something that once we learn how to do it, they will have better yields. Right now, they don't. Right now, they're absolutely abysmal in terms of metabolic yields, just because they're so primitive, there's no robustness in them. But that's just very beginning. It's only been the recent few years when we are able to do it. So hopefully, as we get better at it, we will be able to build systems that have better yields. And other people are working on it. For example, there's work from Mike Jewett's lab where they are trying to make better ribosomes that are faster, ribosomes that are more efficient. And all of that definitely helps us hope that one day synthetic cells will have better yields, better efficiencies.
Yeah, so most of the components that you mentioned, they are kind of slow in this kind of synthetic systems. But is there something like reverse, or did you notice that something is faster than the living systems, for example, like some sort of component which is working much better in the in vitro system than in the living system?
Certain metabolic processes are actually easier in vitro. Some enzyme pathways that are normally slowed down because of the nutrients are being diverted to other pathways If you reconstitute it in a simpler in vitro system, they speed up, they get more efficient, but it's relatively rare right now. There are very few pathways like that.
So the last thing is like in this initiative that you've been part of, Build a Cell, where it's an interesting, I think, setup in a way, because I always think of physicists where they are collaborating, I mean, at like this large facilities to do something like to work on this huge projects. For example, the best example is CERN, where the fascinating thing was that last paper that I saw from them, it was like with 5,000 authors or something.
Yes.
I mean, that's just like height of collaborative effort in science, right? And when I see this kind of initiatives, like the kind of initiative that you are coordinating as well, Build a Cell. So what is it and how it can help in actually building a cell?
So the science has to be done collaboratively. And physicists obviously figured it out because they need those giant investments like the Hadron Collider. It's not possible for a single country to build that. It has to be international collaboration. It's much harder to imagine in biology what can be done like that, because we don't need a giant investment, single investment like that. But we still need collaboration. We still need to bring all the people together. And that's what a Build a Cell is. It's an international collaboration of people that want to build synthetic cells and want to use them for all the practical applications. We all understand that in order to do that, we have to be collaborative. We have to work together. And that's really what drives that whole collaboration and what drives the field, is bringing people together and figuring out how can we work together across all of those boundaries, geographical boundaries, discipline boundaries. How, for example, me as a chemist can work with someone who's a physicist and with someone who's a biologist on putting together this incredibly complex living cell. And Build a Cell is a community, but it's also a research collaboration. We meet, we share ideas, we share data, we help train students, we help train people that one day will start their own labs. And we also work on outreach and education. We talk to the public about this field, about doing this work, why it's important, what can it bring to people, what can it give people. And to use that physicist analogy, we don't need a single facility. Like, we don't need a Hadron Collider, we don't need a space telescope. There is not a single building that you could build and everyone goes there and builds a cell. That's not needed. But what's needed is this international community working together, sharing ideas and working on this one problem that we're all sharing. And that's what Build a Cell is that it helps us come together and work together as one community, despite being spread all over the globe.
Yeah, fascinating. I mean, I could ask for the agenda, but let's put it in different ways. So, what are the major questions according to Build a Cell community regarding synthetic cells?
The main question is how to build the damn thing. We still haven't done it. We're trying to share expertise and figure out how to overcome all those problems in building cells. So how to make a cell replicate, how to make all the components of the metabolism replace each other, how to put together all of those systems that we need in order to make a cell. And that's the biggest driving force behind our work.
Yeah.
There's also more practical questions. For example, how do we fund international collaborations? How do I get my collaboration with someone who's in the Netherlands and someone who's in Japan funded? There are very few mechanisms that can fund international collaborations like that. And that's another thing that Build a Cell is trying to do, is figuring out how to fund international collaborations like that.
Yeah, and I think it would also depend on, again, if there are some bridging grants, etc., right? Which, yes, I think it's rare.
It's rare, and it doesn't just spontaneously happen. That's part of the advocacy that we're doing, is trying to convince people that this is something that needs to happen and that is worth happening.
Yeah. And so what do you think, how far in terms of the complexity you would like to go when you're thinking of the synthetic systems? Because again, is it like the aim is to, or do you think we'll have to meet like we'll have to meet this top down approach, like in that way we'll have to meet to those guys, or we can kind of simplify biochemistry in a way and stop, I don't know, a bit earlier and see what is the minimalistic system in a way?
That's a $1 billion question, because if we knew the goal, we would just go straight there. Right now, we don't know how much complexity we will have to build for this to really be living. And to me, the ultimate goal is a cell that can self-replicate and evolve on its own. And my guess is that the complexity will be about halfway between what we have right now and what the mycoplasma minimal cell is. But we're not going to know it until we actually build it. So it's all kind of speculations at this point. But yeah, to me, an ultimate goal is a living synthetic cell.
Okay, great. So at the end, can you please suggest some like reading material? What are or did you read any recent book on the topic which people can read, etc., or some papers, I don't know, some researchers to follow?
There's not many books specifically on this topic. It's still a relatively new field. But if you look up the Build-A-Cell website, every person that's listed there as a Build-A-Cell PI has their own website. That's pretty much the thing that people do these days, is every lab has their own website and lists all their papers there. And there is a lot of outreach that we're doing as well. So if you look up on the Build-A-Cell website, we have a whole page on basically synthetic cells 101, like how to build a cell, how to get started. And there are references there. So the way we wrote it is that it helps people at every level of education to find something that's accessible and readable at their level. And that's something that I would recommend if someone wants to get started, is just read websites. Don't jump into primary research literature. Just read those explanations on the website. And once you do that, then you'll be able to kind of build up the background and go deeper and deeper.
Well, I did listen to some of those talks and they are fascinating. You see how much progress is there and what kind of difficulties people are facing, and like even at smaller parts. Yeah, so that was interesting. But it's a great initiative, and thank you for accepting the invitation, for doing this and sharing your impressive work, ideas, and of course, building a community about building a cell. Thank you so much.
Thank you so much. Thank you so much for having me. That was fantastic.
Thank you.